For my biology role model project, I chose Cornelia Bargmann, also known as Corey Bargmann. Bargmann grew up in a very academic household. She knew from a very young age that she had a passion for science. She especially loved the hands-on nature of science. She attended Georgia University for her undergraduate degree in biochemistry. She began working in a lab there and eventually was introduced to the genetics department. She worked in Sidney Kushner's lab and studied bacterial genetics and RNA metabolism. She then went to MIT for her graduate degree and studied oncogenesis in Robert Weinberg's laboratory. This is where she began her thesis research on the new oncogene. During her free time, Bargmann likes to play the piano and sing. She began playing at a young age and continues to this day. She also loves to read and learn. Bargmann is currently married to Richard Axel, who is a neuroscience professor at Columbia University. I chose Corey Bargmann because I admired her academic drive. During the time that she went to college, she said that only 20% of students were pursuing a higher education. She didn't let this stop her from continuing her education as she went on to complete her undergraduate and postgraduate degree. I also chose Bargmann because I relate to her. For starters, she is a woman who has accomplished so much, and that is inspiring to women all over the world. I also share her love for science. I am very excited to keep learning and discovering like she has done. Some of Bargmann's most recent research is her work on the dissection of neuronal gap junction circuits that regulate social behavior in Caenorhabditis elegans. In this research, Bargmann worked with other scientists in order to isolate the UNC9 gap junction protein in a transparent worm in order to see the effect that the protein has on the worm's behaviors. The figure shown is a figure from the research article that was written. The top two diagrams show the structure of the gap protein. The bottom three diagrams show the effects that the protein gap has had on the social behaviors of these worms. Some of Bargmann's most important discoveries include her identification of the SAX3 gene, which explains axonal development in depth, and her work on the SYG1 and SYG2 gene. She concluded that these genes work together in order to connect synapses to each other. Bargmann is also continuing her research on C. elegans and their behaviors. My sources are shown on this page. Thank you.